Hello everyone, welcome back to my channel. Today we are going to do a very simple but beautiful microscope experiment. We'll take an onion, remove its thin skin and observe it under the microscope. This is a fresh onion. Inside the onion there is a very thin transparent skin between the layers. The skin is made up of plant cells and these cells are large, clear and easy to observe under microscope. This is a lens cleaning tissue paper which I am using it to clean my glass. Now I am separating one onion layer. Between two layers you can see a shiny thin membrane. This is the onion epidermal peel. This is what we need for microscopy. Using my fingers, I gently pull out the onion's skin. You can see how thin and transparent it is. This single layer contains hundreds of plant cells. I place the onion skin flat on the slide and I add one small drop of water. This helps the cell stay hydrated and clear. Now I gently place the cover slip on top. I lower it slowly to avoid any air bubbles. Air bubbles can disturb the microscope image. Our slide is now ready. This is placed under the microscope. I am starting with low magnification. This is now a focused image. Now the cells are becoming visible. Look at this beautiful pattern. These rectangular shapes are plant cells. The thick lines you see are the cell walls. This is why onion cells are used in biology classes worldwide. This is right now what 100x. You can see the cell walls and even you can see the cell nucleus. You can see how clearly they are in a pattern arranged in a beautiful manner. Now this is magnified much more. This is close to 450x. If you enjoyed this experiment, please like the video, subscribe for more simple and fascinating microscope videos. Thank you for watching.